Today, we're really focusing in on how drugs interact with the central nervous system.
Hmm.
Think of this as a targeted exploration, especially valuable for those of you following KMR advice, navigating your medical studies.
Yeah,
Our core mission today, it's to properly break down the fundamental ways these medications actually work inside the brain.
precisely. And our main resource here is Fundamentals of CNS Drug Action. We're aiming to dissect what can seem, pretty
Very
intricate
complex stuff.
into clear, digestible components, much like you'd want from a really good lecture, right?
Okay, let's dive into those foundational principles then.
Yeah.
Starting with neurotransmitters, the brain's chemical messengers, like chemical emails zipping between brain cells. And drugs often work by, well, tweaking these messages when things aren't quite right.
And what's really amazing is how specific they are. Each neurotransmitter has its main job, and getting that is key to understanding the drugs. Let's start with dopamine.
Dopamine. Often hear about it with pleasure, the feel-good chemical. But it does more, right? Especially thinking about how these drugs act.
Oh, absolutely. Yes, it's central to reward pathways, but it's also absolutely vital for controlling movement and even cognitive things like attention planning.
Okay.
So, thinking about drug action, take conditions where dopamine signaling is, let's say, overactive, maybe causing hallucinations. Certain antipsychotics, like haloperidol, come into play.
And how does that work? What's the mechanism?
Haloperidol acts as a dopamine receptor antagonist. Think of it like it sits in the docking station where dopamine normally fits.
Ah, okay.
By blocking those receptors, it stops too much dopamine from getting its message across. If you've got that overload causing hallucinations, this blockade helps kind of quiet things down.
So it's blocking the connection, not just lowering the amount. But then for something like Parkinson's, where there's not enough dopamine causing movement problems,
Yeah.
we use levodopa. How's that work?
All right, different strategy. Levodopa is basically a precursor molecule. The brain can take it and turn it into dopamine. So you give levodopa, it gets across the blood-brain barrier, gets converted, and boom, you've boosted dopamine levels where they're needed, like in the basal ganglia for movement. It provides the raw material.
That makes a lot of sense. Okay, let's switch gears. Serotonin, or 5-HT, what are its main jobs?
Serotonin has a really wide range of effects. Mood regulation is a big one, obviously. Sleep patterns, too.
Yeah.
And even activity in the gut.
Oh, yeah, the gut connection. That'll come up later with side effects, I bet.
Definitely relevant, yeah.
And a really common drug class targeting serotonin is SSRIs, like sertraline. For mood disorders, how do they do their thing?
So SSRI means selective serotonin reuptake inhibitor.
Yeah.
You've got the synapse, right? That little gap between neurons.
Mm-hmm.
Normally, after serotonin sends its signal,
Mm-hmm.
it gets sucked back up by the neuron that released it.
Yeah.
Reuptake.
Right.
SSRIs block that vacuum cleaner, essentially. They inhibit the reuptake.
Okay.
So by stopping it from being reabsorbed so quickly, you end up with more serotonin hanging around in that gap, the synaptic cleft. This gives it more time to interact with the receptors on the next neuron, amplifying the happy signal, as the source puts it.
And the source mentions it can take weeks to really notice a difference, calls it refilling a battery. Why the delay? It's not instant.
That's a really important point. That analogy is good because it's not just flicking a switch. Increasing the serotonin sets off a whole cascade of downstream changes in the brain.
Like what kind of changes?
Things like changes in how sensitive the receptors are, strengthening connections between neurons, neuroplasticity, basically.
Oh, okay.
And these adaptations, they just take time to happen. That's why the mood effects aren't immediate.
Okay, that clarifies the timeline. Next up, GABA. The source calls it the brain's brake pedal.
Yeah, that's a great way to put it. Very intuitive. GABA is the main inhibitory neurotransmitter. Its job is to dampen things down, slow neural activity.
So it makes neurons less likely to fire.
Exactly. It's the primary calm down signal in the CNS.
And how do anesthetics like propofol fit in with GABA?
Propofol and other general anesthetics work by seriously boosting GABA's effects. They basically put that brake pedal to the floor.
Whoa.
Supercharging it leads to a massive reduction in overall brain activity, and that results in the state of unconsciousness you need for surgery.
A really profound way to hit the brakes then. Now, the flip side, glutamate,
Yeah.
gas pedal.
Right. If GABA slows things down, glutamate speeds them up. It's the main excitatory neurotransmitter.
Yeah.
Crucial for things like learning, memory.
Yeah.
Fundamental stuff.
And the example drug here is ketamine, which has some unusual effects. How does it interact with glutamate?
Ketamine works by blocking a specific type of glutamate receptor, the NMDA receptor.
Okay.
And what's interesting is that disrupting glutamate signaling in this specific way can cause that dissociative state feeling detached. It also has pain-relieving effects.
So blocking the gas leads to dissociation and pain relief? That's complex.
It is. The exact downstream effects are still being figured out, but it's definitely a different mechanism than just broadly boosting or reducing a signal.
Fascinating. Okay, so we've covered the messages, the neurotransmitters. Now let's talk about the locks they interact with the receptors.
Right, receptors. These are specialized proteins, usually on the surface of neurons. Think of them as complex 3D shapes, not just simple locks. They have specific binding sites, like a little groove where only the right shaped key, the neurotransmitter, or a drug can fit and trigger a response inside the cell.
And the source breaks these down into agonists, antagonists, and partial agonists. Let's start with agonists, the copycat keys.
Exactly. Agonists bind to the receptor and activate it. They mimic what the natural neurotransmitter does. Morphine is a classic example.
How so?
It binds to the same opioid receptors that our natural endorphins use, and that triggers pain relief, euphoria. It copies the effect.
So it fools the receptor. Okay, what about antagonists, the broken key?
Antagonists also bind to the receptor. They fit in the lock, but they don't activate it. Instead, they just sit there and block the natural key, the neurotransmitter, from binding.
They jam the lock.
Pretty much. We mentioned haloperidol earlier, that's an antagonist. It blocks dopamine receptors to stop psychosis symptoms. The goal is to prevent the signal.
Right, blocking the message. And then there are partial agonists, the sticky keys. What's their deal?
Partial agonists are interesting. They bind and activate the receptor, but they only produce a partial response, less than a full agonist would, even if all receptors are occupied.
So like turning the volume up halfway, not all the way?
Kind of, yeah. Aripiprazole, an antipsychotic, is a good example. It acts as a partial agonist at some dopamine receptors.
How does that help?
It provides a bit of dopamine activity, enough to help with some symptoms, maybe, but not so much that it causes major side effects like a full agonist might, or the issues from completely blocking dopamine like a full antagonist. It's about finding a balance.
That balancing act seems really key for complex conditions. Okay, messengers, locks. Now the roads these signals travel on. Brain pathways.
Yeah, just like a city has specific highways for different traffic, the brain has these organized pathways connecting different areas, each handling specific functions.
And drugs can target these specific roads.
That's often the goal, yeah, to be more selective.
First one mentioned, the mesolimbic pathway. What's its main function?
The mesolimbic pathway is famously known as the brain's reward circuit. Big role in pleasure, motivation, and heavily involved in addiction. Dopamine is the main player here.
Okay. And how do antipsychotics interact with this pathway?
Well, in conditions like schizophrenia, you can have too much dopamine activity in this mesolimbic pathway. That's thought to contribute to positive symptoms like hallucinations, delusions, those false signals.
Right.
So antipsychotics work by reducing dopamine activity here, aiming to quiet down those symptoms.
So targeting that reward highway helps with psychosis. Okay. Next, the nigrostriatal pathway. Function.
Nigrostriatal is all about controlling voluntary movement. It connects the substantia nigra in the midbrain to the striatum. Crucial for motor control.
And guess what?
Dopamine again. It's dopamine again. Key neurotransmitter here too.
And this brings us back to Parkinson's disease, doesn't it?
Exactly. In Parkinson's, those dopamine-producing cells in the substantia nigra start to die off. Less dopamine in the nigrostriatal pathway.
Which causes the movement problems.
Precisely. The tremors, the stiffness, the slowness, bradykinesia. Levodopa works by boosting dopamine in this specific pathway to help ease those motor symptoms.
Restoring the traffic flow on the movement highway improves function.
Got it. Third pathway, the Raphe nuclei. What's its role?
The Raphe nuclei. These are clusters of neurons mainly in the brainstem. They're the brain's primary serotonin factory.
Where serotonin is made.
Yep. And neurons from there project out to many, many brain regions involved in mood, sleep, appetite, lots of things.
So this is where SSRIs are acting again.
Well, SSRIs increase serotonin everywhere by blocking reuptake. But the source of that serotonin, the regulation point, is largely the Raphe nuclei. Boosting the signal originating from there helps lift mood over time, that refilling the battery idea again. Okay, and the last one, the thalamocortical loop. Function.
This is a really complex circuit. It's the back and forth communication between the thalamus, the brain's sensory relay station, and the cortex, the outer layer. doing the higher thinking.
What does it do?
It's vital for filtering all the sensory information coming in and generally regulating how excitable the brain is.
And anti-epileptic drugs target this loop. How?
In epilepsy, you often have instability in this thalamocortical loop.
Yeah.
It leads to those bursts of abnormal, excessive electrical firing seizures, the electrical storms.
Right.
Anti-epileptics use different mechanisms, but the goal is often to stabilize this loop, make the neurons less likely to fire excessively, and basically prevent those storms from happening.
Regulating the electrical flow to stop the seizures. Okay, wow, we've covered neurotransmitters, receptors, pathways, a lot of ground. But now, the crucial part.
Side effects.
Why do they happen?
Yeah. The million-dollar question in pharmacology. The key thing is, drugs are rarely perfectly targeted, meaning they often hit more than just the intended receptor or pathway. They might interact with similar receptors elsewhere, or affect systems you didn't mean to target. That lack of perfect specificity leads to side effects.
Okay. And the source gives examples. Let's start with antipsychotics causing stiffness. Why does that happen?
Okay. So remember, antipsychotics block dopamine receptors, right? They do that in the mesolimbic pathway to help psychosis.
Yeah.
But dopamine is also critical in that nigrostriatal pathway, the movement one.
Ah, okay. I see where this is going.
Exactly. The drug isn't perfectly selective. It also blocks dopamine receptors in the movement pathway. Result, dopamine deficiency there, leading to muscle rigidity, stiffness, like the muscles get stuck in park.
So the helpful mechanism in one place causes problems in another because dopamine is used in both. And the fix mentioned is anticholinergic drugs like trihexyphenidyl. How do they help?
It gets a bit complex, involving the balance between dopamine and another neurotransmitter, acetylcholine, in those motor areas. But basically, anticholinergics block acetylcholine. Reducing acetylcholine activity seems to help restore some balance and counteract the stiffness caused by the low dopamine activity.
Shows how interconnected it all is. Okay, next example. Nausea with SSRIs. Why that specific side effect?
This is a great example of a brain drug affecting the body. SSRIs boost serotonin in the brain for mood. But guess where most of the body's serotonin is?
You mentioned the gut earlier.
Bingo. About 90% is in the GI tract.
Wow, 90%.
Yep. So when SSRIs increase serotonin levels systemically, they also increase in the gut. This can overstimulate certain serotonin receptors there, particularly the 5-HT type, which are known to trigger nausea and vomiting.
That makes total sense why gut issues are so common at first. And the advice is take with food or it fades in 1-2 gets.
Taking it with food can slow absorption, maybe lessen that initial gut serotonin surge.
Yeah.
And yeah, often the body adapts. The gut receptors might become less sensitive over time, desensitize, or other gut mechanisms compensate. So thankfully, it often gets better.
Good to know. Okay, last example. Memory fog with benzodiazepines. What's causing that?
Benzos work by enhancing GABA, the brake pedal.
Right, calming effect.
Exactly. But if you enhance GABA too much in the hippocampus, a brain area absolutely critical for forming new memories, you slow down the processes needed to encode and store that information. Result, that feeling of mental fuzziness or memory fog.
So the calming effect spills over and dampens memory formation. And the fixes are lowering the dose or using shorter-acting benzos like lorazepam.
Makes sense, right? Lower dose means less overall GABA enhancement, less impact on the hippocampus. Shorter-acting drugs clear out of the system faster, so the effect, including the memory effect, doesn't last as long.
Less time with the brakes slammed on the memory center. Got it. Now, the source had that handy takeaway table summarizing all this.
Yeah, that's a really useful quick reference. Drug class, target, good effect, side effect, and why the side effect happens. Connects all the dots.
Definitely. So just to quickly recap our discussion today, we've looked at neurotransmitters as the chemical messengers,
the signals.
Receptors as the specific locks or docking sites,
Locks and key.
brain pathways as the highways these signals travel on,
The specific routes.
and crucially, why side effects pop up, because drugs aren't always perfectly precise and hit unintended targets.
Absolutely. And having a solid grip on these fundamentals, well, it's essential if you're going into medicine. It's the foundation for understanding current drugs and, importantly, developing better ones in the future.
Which brings us to a final thought for you listening, especially on KMR Advice. Given how incredibly interconnected all these brain systems are, what do you think are the biggest challenges, but also the most exciting future directions for creating CNS drugs that are even more targeted with fewer of these off-target side effects?
It's a huge question.
Definitely something to chew on as you continue your studies. How do we get even smarter about hitting just the right target in just the right place at just the right time?
Indeed. Neuropharmacology keeps moving fast. Understanding these core ideas is really the key to being part of those future breakthroughs.